uh, getting the recording going. But with that, I would like to leave it to David. So take it away.
All right, thank you, Dana, and welcome everybody. I appreciate you showing up this morning. Well, for me, it's this morning. I just got to my office. It's 8 o'clock here. I've got to teach a class at 10, and so I'll get this done. So this is my first time doing one of these things, and I went back and forth with Dana and Susan a little bit on what this thing should encompass, and I really didn't know. I found out about the GBATnet YouTube website this morning, and I saw there was a bunch of talks like this on there. I sure wish I had looked at some of them, but I didn't because I just found out about it. So I apologize if this is too complex for you. I also apologize if it's not complex enough for you. I tried to throw in a bunch of stuff from the very simple to the more complex so that I could hit everything here. So what is genomics and why should I care? So obviously I'm part of BAT1K, and our objective is to sequence and assemble the genomes of every extant bat species over the next several years. We've gotten a good start already. We're well on our way. We have one representative from every family, and we're working on getting representatives from other ones. One of the suggestions Susan had when we were going back and forth with my talk was to talk about, just some basics. What is the difference between genetics and genomics? I'm assuming that some of you have at least explored genetics a little bit, either in college or high school or some other educational experience. And in genetics, you would have learned about alleles, and you would have learned about Punnett squares and all that kind of thing. But typically, genetics is focused on isolated parts of the genome, this gene or that gene, or the inheritance of different characteristics as they go from generation to generation. Genomics is typically more broad in that it encompasses the entire genome and looks at the structure of the genome, the way that the different parts of the genome interact with one another, the way that the genome interacts with the environment. It just basically is a larger scale aspect of genetics. It takes into in consideration a lot more, a much broader range of topics than does genetics typically. So obviously, if we're going to talk about genomics, we have to talk about what a genome is. There are various definitions of a genome, and the definition of a genome for every individual organism is going to be unique. But a generic definition of a genome would be the minimum nucleic acid complement that defines an organism or an organelle or a virus. And of course, as I said, this is going to vary depending on the type. For example, most organisms have DNA for their genomes, but there are some viruses that have RNA-based genomes. A genome can take lots of different shapes. Most eukaryotes have genomes that consist of linear chromosomes. Other organisms like bacteria, most bacteria or many bacteria, have circular genomes that consist of one circular not capsid, but ring of DNA. But they can also have plasmids and all sorts of things. There are basically as many different types of genomes out there as you can imagine. Some organisms and some viruses rather have genomes that are made up of both DNA and RNA. So it just depends on what organism you are as to what your genome will consist of. But for the most part in this talk, because we're all interested in bats and bats are eukaryotes, I'm going to be thinking mostly about eukaryotic genomes, and in particular, especially when I get to the end of this, bat genomes in particular. So you'll often hear that the genome is a blueprint. I hear this all the time. I listen to a lot of debates, and debates on the origin of life, debates on religion, things like that. And very often the argument is made that DNA is too complex to have evolved on its own because it's a blueprint. Well, it's not a blueprint. It isn't. It's a chemical. It's a bunch of DNA, and that DNA has chemical activity because it's a chemical, and it interacts with other chemicals, proteins mostly, and those proteins then go out and do certain jobs. But it's the way that that DNA chemical is arranged that allows for that to happen. Basically, what the DNA does is it facilitates a growth of an organism by not really directing, but containing the information necessary in order to tell the cell how to generate the different proteins the cell needs in order to go about its business. It doesn't dictate who you are. However, it does provide information on what you can be. But that information is part of a system. And I mentioned in that definition up there, it's a chemical that interacts with the environment because that's what it does, right? You have information and stimuli coming in from outside the cell that will interact with the DNA and therefore influence what it does. For example, that the bottom picture there, that's Shaquille O'Neal. He's a famous basketball player here in the States, and he's very, very good at what he does. And he's a giant. He's just a huge, huge person, and he has a lot of skill. But if you can imagine, a lot of that skill was inborn because of his genetic code. But if he had, when he was young, not been given access to the proper nutrition, if he had not been given access to the proper training, if he had not been given access to the right exercise, he would not have become the athlete that he is. So the DNA does not dictate what you will be. It in some ways restricts it, but it also facilitates it in becoming that. And the way that I always talk about this in my class is if someone were to clone you, you would not end up being the same person that you are today because you wouldn't have encountered a different environment because you were born 20, 30, 40, or in my case, 55 years ago. And the world is just different. And I would encounter, that clone of me would encounter a different environment, which would end up producing a different person. So genomics is a relatively recent scientific endeavor because a lot of the tools that we have that we need to use to investigate genomes did not exist 50 years ago. It was only 50 years ago or 70 years ago now that I think about it, that we just figured out what DNA was and how it was put together with Watson and Crick. And the development of remote robotics and automated sequencing, but most importantly, computational methods have facilitated the development of this field. But before we get started, one of the things I want to talk about is this, it's a pet peeve of mine whenever I'm talking to people about genomes and in genomics, is I want to make sure that everybody understands the difference between a genome and a genome assembly. Most of the time in genomics, we're mostly dealing with genome assemblies, and that is different from a genome. We can interact with a genome. We can manipulate and experiment with genomes. And a genome is a physical thing. It is a thing that exists inside the nucleus of an organism, if it's a eukaryote or in the bacterial cell. But it's a thing. It's made up of DNA typically. But a genome assembly is an approximation of that genome, and it's made up of computer code, basically bits and bytes that exist in a computer. But because genomes as DNA are small and difficult to manipulate, a lot of times what we do is we use these genome assemblies as proxies for that genome, and we investigate those instead of investigating the genome directly. And that is what I'm going to be, that's what my lab does primarily. We have done some genome manipulations as well, but most of the time we're dealing with genome assemblies. And so obviously that's where I'm going to spend a lot of my time focusing. So this field has expanded dramatically over the past 20 years or so into lots and lots of different subfields, including applied genomics, where you can manipulate those genomes. As I mentioned before, there are things like CRISPR, where you can take and replace this gene with a modified version of that gene or knock out the gene altogether. Population genomics, which we do a lot of in my lab, where we look at markers from across the entire genome to investigate what the dynamics of populations of bats are. Regulatory genomics, in which you figure out what parts of the genome are regulating genes and causing genes to be turned on or turned off or upregulated or downregulated. Structural genomics, it was surprising to find a few years ago that the genome is not just mixed up completely inside a nucleus, but there are different domains in which chromosome 1 will find itself, and chromosome 2 will find itself, and chromosome 3 will find itself within the genome. It's actually a very well organized situation inside the nucleus, not just a jumble of DNA threads. Bioinformatics, which is the computational applications to genomes. Metagenomics, you probably heard of. This is where you investigate the different organisms that say make up your gut microbiota, or the metagenomics of, I think Craig Venter many years ago spent time going out at sea and collecting all the different microbes that you might find in a teaspoon of ocean water, et cetera. Functional genomics, disease genomics, comparative genomics, and phylogenomics. And these three that just I highlighted in red here, these are the three examples that I plan on giving later on, where I give examples of how genomics can be applied to different questions that we might have in biology. So what do genomes look like and how do we make sense of it? This is what a genome looks like when you look at it on, or a genome assembly, I should say. It actually is made up of A's, G's, T's, and C's, unless you're dealing with an RNA genome, which we're not going to really talk about here. And this, of course, is a very small snapshot. What I did last night when I was working on this was I pulled up the genome for Eptescus fuscus on my computer. And this is what looks like if I use the command cat, which just causes the genome assembly to just flash across your screen, line by line. And this is what I found. And as I was doing that, I thought, okay, well, how long would it take to go through the entire genome if I were to do this and, at this rate of speed? So I assumed that maybe 30,000 base pairs were going by every second. And I did the math and it came up to 27 hours. It would take 27 hours for that entire genome to go by. That's about 2.5 billion base pairs going around, across my screen at that rate. And so it's huge. These things are massive. This is why it is the development of computational infrastructure that has allowed us to investigate genomes more efficiently over the last several years. We simply couldn't have done this with a Commodore 64, which was my first computer in the 80s, because the computational power just simply wasn't there. So that's what a genome looks like, or at least a genome assembly looks like. And we use that to investigate the actual genome. So what is a genome made up of? This is a figure that I used to use for my cell biology class when I was talking about the construction of the genome. And this is, of course, a representation of the human genome. And be aware over there on the left side of the screen, it says, these are the different parts of the genome that belong to different categories. And I'm mostly talking about here eukaryotes like us and bats. So you'll see that the genome is made up of a whole bunch of different parts. That part all the way to the right there that says approximately 45%, that's actually the part that I'm most interested in as a researcher. It's made up of repetitive elements in the genome, parts of the genome that you find repeated over and over and over again. For example, in the human genome, there's this DNA sequence called ALU, which in all of our genomes, we have about 1 million copies of. They are nearly identical to one another, and they are scattered essentially randomly around the genome. On the opposite side, the other 44%, that is just stuff. It's old stuff that probably serves no purpose whatsoever in our genome. You can get rid of it, and it doesn't seem to affect the function of the genome in any way whatsoever. Down there at the bottom, that 6.6%, that's heterochromatic DNA, basically the centromeres and telomeres and the end caps and the middle parts of many chromosomes that are just highly repetitive stuff that is condensed and inaccessible by most proteins. The parts that most people are interested in, not me, but many other people, are that 1.5 and 3% right up there at the top. That 1.5% represents the actual protein coding genes of the genome. So it's a really actually tiny part of the genome that actually encodes for the proteins that most people are interested in. That other 3% are the regulatory sequences, mostly, that are determining when those coding genes get turned on and turned off. So the actual functional parts of your genome represent, at least most big eukaryotic genomes like ours are actually a really small part of what makes up a genome in particular. And that's going to become really, really important later on when it comes to talking about how we end up getting these genome assemblies put together and figure out what they're made of and what they consist of. Real quickly, just a definition, because I'm going to use these terms later on in the talk. I've already used the term gene. This is just a region of the genome that encodes a protein or encompasses some part of the DNA that does something. It encodes some regulatory sequence, or it encodes a non-coding RNA or something like that. Basically, it's a locus that does something. And of course, I just used the term locus, which is the bottom one down there. That just means a place. It's an address in the genome. So I live at 5806 76th Street in Lubbock, Texas. Hopefully nobody will take that and send a bomb to me or anything. But that is my address. And that we can use that same term to figure out where something is. We say it's on chromosome 1 at position 11,576,812. And of course, we also have alleles. This is something you probably learned about in any genetics class that you have. These are just alternative forms of a gene. So, I got 2 genes. from one from my mom and one from my dad at whatever locus, and they may be the same or they may be different. And if they are the same, they are the same allele. And if they are different, they are different alleles. And so it's just a matter of different versions of some locus that you might find at that locus or that place in your genome. So here example, it is an example of how we end up trying to look at these genome assemblies. We try to look at them in terms of where are the genes, where are the regulatory sequences, where are the transposable elements, the repetitive regions of the genome. This picture just shows different parts of the genome. That red part right there at the top, that is a coding sequence. That one at the bottom is as well. This is some gene that leads to the production of some protein. Those other parts that are being pointed out, those are regulatory motifs, things like the TATA box, which is the start point for, or the binding point for RNA polymerase, which would then give rise to an mRNA that would then be translated into the protein. But you can see that trying to figure those things out, we're only dealing with 4 letters here, and they look pretty much random if you were just to look at this thing. Trying to tease out what those bits and pieces are is actually kind of difficult, because you're dealing with so much information over such a broad amount of information, and trying to tease that information out can be relatively difficult. But again, that's where the computational biology and bioinformatics comes in, that we've been working on for so long over the past 30 years or so, 40 years. So more information about what genomes are. I think it's important to point out that there is no one genome sequence for a species. We are working at Bat1K to generate one genome sequence or one genome assembly for every species. But that does not mean that that one genome assembly represents the totality of the genomes of that entire species. Because if it did, then all of those species would be clones. And we know that they're not. We know that there is variation between individuals or among individuals in any species. And so we have these variants. There are differences between me and you. Several million base pairs differ between me and any of you that are in the audience. And so it's that variance, those variants, that are actually where the difference is in the way that we express our genes, and the way that, I'm bald. Most of you are probably not. You probably never will be. But I have variants in my genome that lead to this condition that I'm unfortunately going to have to deal with for the rest of my life. And so it's that variation that is important, and that's what we're trying to get to when we're doing these investigations into genomes. So real quickly, I don't want to spend too much time on this, but genomes vary vastly in size. As you can see right here, here's us, these mammals. Our typical mammal genome is anywhere between 2 and 4 gigabases, 2 and 4 billion base pairs long. Reptiles about the same size, birds slightly lower. But look at these things like protozoa. Some amoeboid genomes are hundreds, or at least dozens of times larger than the human genome, than the mammalian genomes. Same thing with some angiosperms. In fact, the largest not eukaryotic genome, but the largest multicellular eukaryotic genome is actually a plant. I believe it's lily, but I can't remember for sure. But they are huge. And that goes back to that idea that there's a lot of the genome that really doesn't do anything. All of these eukaryotic genomes all contain about the same number of genes, about 20,000 genes, sometimes more if there's been some sort of a duplication. But they all contain basically the same genetic complement as far as the genes are concerned, because they all have to do basically the same things: take in nutrients, metabolize those nutrients, and do all this basic stuff, replicate their DNA and grow. But there is all this extra stuff in there that really doesn't do anything, at least in most eukaryotic genomes. And so one of the things that there's this what's called the C-value paradox, this idea that genome size is not related to organismal complexity. And that is something to keep in mind as you're talking to people about this sort of thing. So going back to a quick lesson from genetics, hopefully if you have taken a genetics course, you learned about the way that genomes are structured, and you learned about alleles and things like that. And what you find is that, if we're talking about humans or bats, we all get our genomes from mom and from dad. We have 2 copies of our genomes, essentially. We have 2 haplotypes. And one of those came from mom, one of those came from dad. And what you're seeing right here are two chromosomes, one of them from your mom, one of them from your dad, and you'll see the different alleles. These are three loci on those chromosomes, and you have the same gene at both positions on mom and dad's chromosomes that you got from them, but they're different alleles. This one has the A allele on all three positions. This one has the B allele at all three positions. And what we'd expect is that those would be inherited as a unit. All three of these A alleles would continue and we would pass those on to the next generation with all three of those A alleles together. And we would pass on all three of those B alleles together since they're on this one linear chromosome. But that does not happen. Instead, what we see is, yes, sometimes we pass on all three A alleles together, but sometimes we end up having the A and the B and the A being passed along together, or the two A's and a B, or a B here and an A there and a B there. And so they end up actually getting mixed up together at some point during our development and during the production of sperm and eggs. And so that actually is, how does that happen? Well, most of you probably already know how that happens. It happens through recombination crossing over. This process in a cell in which the chromosomes will line up together and then exchange bits and pieces of one another. And it happens that the farther apart those two loci might be, the more likely it is that recombination will occur and mix up those alleles. And the closer they are together, the less likely that is to occur. And we can use that information in order to investigate genomes. In fact, this was one of the very first types of whole genome investigations that was actually done. It's this idea that if you can figure out how often recombination occurs between phenotypic, visible traits that you might see in an organism, we call those phenotypes, that you could figure out what the organization of those genes were on those chromosomes. So if you're seeing a lot of recombination occurring between A and B, and those A and B show different physical characteristics that you can track, then you know that those two genes are far apart from one another on a chromosome. And if you don't see a lot of recombination, you know that those two genes are close together. If you take all that information together with all the other genes that you might be seeing, all those other characteristics you might be seeing, you can then generate a map of a genome. And the first of these maps was actually developed way back in the early 1900s by Alfred Sturtevant, an undergrad in Thomas Hunt Morgan's lab. And he generated the very first genetic map of Drosophila, this model organism that we use all the time now. Now, the problem with these, excuse me, physical characteristic-based maps, phenotypic maps, is that there's relatively few genes. Of those 20,000 genes that we have, relatively few of them actually produce phenotypes that you can see with your eyes. So later on, as we got better and better at manipulating DNA and understanding what DNA was made of, we started using different maps where we could get finer scale maps by looking at things that were closer together. Things like restriction digest or restriction enzyme targets, which we don't need to get into. This is the only time I'm going to mention this. Short tandem repeats or microsatellites. These are the loci that crime investigators use in order to get a DNA fingerprint. AFLPs, rapids, all these are different terms for different markers that you can use that give you a finer scale genetic map than using those phenotypic or visible characteristics that you might see. But what we eventually want to get to is the finest scale map that we could possibly get, the ultimate map. And that ultimate map is what we're dealing with here, getting a genome assembly, knowing the identity and position of every nucleotide, every A, T, C, and G in the genome that we happen to be investigating. And there are two steps to go about doing that. One is to sequence the DNA, which is relatively cheap and easy. It hasn't always been, but nowadays it is. And then assembling the fragments, getting those puzzle pieces together. And we really are dealing with a puzzle here, and putting that together into an entire genome assembly. So how do we do this? This is what I'm going to spend a little bit of time talking about. We do it by using sequencing, DNA sequencing. And early on in the mid-1970s, a guy named Fred Sanger came up with this chain termination-based sequencing, which was the state of the art for 20 to 30 years. This is what everybody was doing. I did it when I was doing my PhD, everybody did it. And basically what it relies on is trying to generate pieces of DNA, but stopping it essentially randomly along the way and getting these pieces. Then you run those pieces through a tube, and then you look and see what the pieces are consisting of through a detector, basically a camera with a laser, and then you use a computer to get something like this that says, okay, AAA, GTC, so on and so forth. And that was the state of the art for a long time. And it worked really, really well. And this is what was used to generate the first human genome assembly that we got in about 2001. But the best case scenario for Sanger sequencing is that you could get reads. And when I say a read, I mean one single stretch of DNA that was sequenced of about 700 to 1,000 base pairs on average. The biggest machine that you can see over here that would run was 96 capillaries. So you could do 96 samples at a time. It took about 3 hours to run. And you could get about, like I said, 700 to 1,000 base pairs per capillary, about 5.4 million base pairs during a day. And at that rate, to sequence one human genome, which is about 3 billion base pairs, it would take 600 days to sequence that genome to what's called 1x coverage. Now, what does 1x coverage mean? Well, 1x coverage just means that on average, you manage to hit every base one time. So you would sequence the equivalent of one human genome, and that meant that you got 3 billion base pairs. And you would think, okay, well, you got 3 billion base pairs. That's enough to sequence the human genome. But is it really? And the answer to that question is 1x coverage enough? The answer is no, it's not enough. And there are several reasons why. And one of those is that, for one, we can't sequence from end to end of a chromosome, so we don't know what we're missing. We have to sequence these things in relatively small fragments. We also typically make our sequencing libraries. These pieces of DNA are going to be fragmented into small pieces, and we grab those pieces randomly. And all of our sequencing technologies are not perfect. They have errors. So essentially what happens is this. Imagine you have a genome, and we get 5 copies of that genome, and we break it up into small pieces, which is what we have to do in order to generate a genome assembly and to sequence those pieces. We fragment them all. All this happens in a single tube. So all those fragments end up getting stuck into this one tube. And at that point, what we're going to do is we're going to start sequencing. But because all those fragments are in one tube, we're going to end up pulling them randomly out. And sometimes we'll get that green one 3 times. Sometimes we'll get that red one not at all. Sometimes we'll get the yellow one 5 times. Sometimes we'll get that other green one 1 time, but there's going to be a problem. If we only sequence to 1x coverage, then we're going to have gaps. We're going to miss this piece, and we're going to miss that piece, and we're going to get many copies of that piece right there. But then that ends up being a gappy assembly, an assembly where pieces are missing. So 1x coverage is not enough. Instead, what we have to do is go to higher coverage. We have to try and sequence things deeper than 1x coverage. And in fact, if we were using Sanger sequencing, we would want to get to about 10x coverage in order to get 100% coverage of our genome. Because if we only go to 1x, we're only getting about 63% of the genome. And that's not good enough. We need to be able to get, you wouldn't want to miss 30% or 35% of your genome if you're interested in all parts of that genome. So typically, if we want to get to 10%, you can imagine how much this cost way back in the 90s when this was first going on, when the Human Genome Project was working. So the cost per 1 million base pairs was about $9,000. That meant a human genome being 3 billion base pairs or 3,000 megabases. It would have cost about $27 million to get 1x coverage of the human genome. To get 10x coverage, in other words, to get to 100% coverage of that genome, we would have to spend $270 million at that time. But that only encompasses the sequencing, and nobody does anything for free, as we all know. So the end cost for that first human genome draft was $2.7 billion, a huge amount of money. So that sort of makes getting genome sequences infeasible if that's the case. Fortunately, clever people came along and found ways to generate what's called second generation sequencing. Sanger sequencing was the state of the art for a while, but when second generation sequencing came along, it reduced the cost significantly. It went down to, you can see it right there, it went down from $10,000 per million base pairs of sequence to a couple cents per million base pairs of sequence. And that's where we are now, which has made this a much more feasible process for getting genome assemblies for any given organism we're interested in. One of those technologies is called Illumina. If you have any interaction with anybody doing genomics or genome sequencing, you have probably heard this term. Illumina is the name of a company. I am not going to go into the details of how Illumina works. That is, if you want to do that, take my class. But for now, it's just be aware that it is a system where you can sequence massive numbers of DNA fragments at very, very low cost. In fact, that cost, if you recall from Sanger, it was about in the 90s, about $9,000 per megabase or something. We're talking about the cost per gigabase here is about $4 per gigabase. So just a tiny reduction or a tiny fragment of or fraction of the cost that it was before. So it's cheap and it's easy. There are drawbacks, however. If you look over here at this figure, what you're seeing, this is a typical read that you might see from Illumina sequencing. And what you find is that these colors over here represent quality scores of the sequence that you're getting. And you see there's a precipitous drop off right about 150 base pairs. It goes up to 250 to 300 base pairs, but you're getting a lot of loss of quality. You're not sure of what base you're getting after a certain point. So typically, a typical Illumina read is only 150 base pairs. Whereas if you remember from Sanger sequencing, we were getting 700 to 1,000 base pairs. And you can imagine that the longer the fragment that you're getting, the better your sequence, the better off you are. There are longer read type sequencing technologies out there. PacBio and Nanopore are two of them. If you're doing anything with sequencing with people, this is something you've probably heard of. Again, I'm not going to go into the details of what those things are, but you'll find that you're getting a lot better sequence. This one goes up to 4,000 base pairs, but typically, PacBio and Nano, you can get up to 40,000 base pairs, way better than Sanger or Illumina. The problem is that they have very high error rates, between 10 and 15%, depending on the technology, whereas Illumina sequencing and Sanger sequencing are 99.9% accurate or more. But again, the cost is still much better than Sanger sequencing. So one of the things, if you eventually start working with Bat1K, or you have worked with Bat1K to help us get the DNA that we need in order to do these things, what you'll find is we have kind of stringent requirements for what we need as far as getting our DNA. We need high quality DNA because what we're doing are these next generation sequencings with sequencing technologies with PacBio and Nanopore. And for that, because we want to get such long reads, we have to have very high quality DNA, the DNA where the fragments or the chromosomes have not been broken up into small pieces. And there's a problem with that because DNA has to be treated very in a particular way in order to generate that. And sometimes that can be logistically difficult, taking liquid nitrogen out into the field, using different buffers to sustain the DNA, and it can be cost prohibitive as a result of those problems. But Bat1K is trying to help people get that done. I'm not going to go through all the figures here. This is, again, something from my class, but just be aware that if we do want to get a high quality genome assembly for your organism, we have to have high quality DNA. So speaking of assemblies, I can't go through this without talking a little bit about how to get a genome assembly. And basically, it's putting together a puzzle. What you see up here at the top, these are two reads of DNA sequence that we would get from either Illumina or PacBio or Sanger or whatever. And this consists of A's, T's, C's, and G's. And same with this one down here. What we do when we're doing a genome assembly is we look for matching DNA sequence in order to link those two reads together. And then we link those together, with other ones, and we end up getting something like this, which, we would then pile up on one another and get a final consensus sequence that says, okay, this is what we think the genome assembly looks like. And here's an example of something like that. I'm not going to go into the difference between single end reads and paired end reads here. Just be aware that these are two different ways that we can get DNA sequence. But this is the basic process that we're doing. We would identify two pieces of DNA that have overlap, and you can see that overlap right there. They basically are the same sequence. These are two reads that are very, very similar to one another. Then we find another set that maybe don't overlap quite as much, but they still do overlap, so we can see that they're similar to one another. We do the same thing with another piece, and we find that, yeah, they're similar to one another. We can put them together. Same thing here. And then we say, okay, well, now we've got these basic overlaps. Can we overlap them with one another? And we can. Say, okay, that GCCG overlaps, or that overlaps with this one. or this one overlaps with nothing by itself, but eventually it will. And you can see there's an overlap right there. And we can generate what are called contigs. So those contigs represent those pileups of these different sequences. And then we can use those other sets of reads, those paired-in reads, to link those together. We can say, okay, well, that one is there. We know there's a certain number of base pairs between the two ends of that read. That matches here. So we now know that those two belong together. And we can see that that goes here. But that one, look, it matches with that CTTAGCC over there. We now know that we can get those matching up with one another. And we can do that over and over again until we get a final consensus sequence that represents all of those reads put together. So it very much is like putting together a puzzle. And that puzzle, in this case, generates that sequence down there at the bottom. Now, you'll notice that these were very, very short reads. These, in this case, were only about 20-something base pairs or so at max. You can imagine, oh, I'm sorry, I meant to tell you this. This is basically that same process, but in reality. This is a bacterial genome assembly that we use in my class that I'm teaching later on today. And you can see here's all those reads that you might imagine. They're all piled up on one another, just like I showed you. But these are short reads. You can imagine that if you had bigger puzzle pieces, it would be easier to solve the puzzle. For example, this is a picture of Myotis thysinodes on an underpass or an overpass in New Mexico. Here is another picture. That's that same picture, but much smaller pieces. You can imagine it would be much easier to put this back together than it would be this one, because these are larger pieces. So that's where these long read technologies come in. You could do this with these short reads, and those circles there represent different errors in the sequencing technology. Or you could use long reads, and you can see that there's much better overlap. So it's easier to put the puzzle pieces together in the case of using long reads than if you were using short reads. The problem, of course, is that trade-off. If you recall, the short read technologies like Illumina have really high confidence rates in what the bases are, but these longer read technologies have lower confidence in what they are. So there's a lot more errors that you have to deal with. So there is a trade-off. So typically most genome assemblies will rely on using two different technologies. Another thing that makes genome assemblies difficult are, the fact that you're dealing with massive, massive amounts of data and uneven coverage, errors that we just talked about, and repetitive regions. Those repetitive regions, which, remember, take up about half of a eukaryotic genome in many cases, lead to misassemblies of that genome. They lead to problems in the way that those genomes are put together. And long reads help with that. So if you are dealing with anybody dealing with genomics, you're going to have to ask them, how good is your assembly? There are different ways to measure the confidence in your assembly. You'll often hear the term N50, which is a measure of how contiguous your assembly is, how big are the pieces that make up your assembly. Typically, the bigger they are, the better. How deep was your sequencing? Typically, the higher, the better. How good do your raw reads map back to your assembly? In other words, how well does your assembly represent the raw data? And also, since most people are interested in genes and the way genes work, there are ways to say, okay, well, do we find all the genes that we expect to find? These things like BUSCO, Benchmarking Universal Single Copy Ortholog, which is a program that we use to investigate whether or not we're finding all the genes we expect to find. So that's how we get to this assembly part, how we get to a genome, as people call it, or a genome assembly. But that's not the point. I spent a lot of time talking about that, but the point of doing all this is to get a genome assembly so that we can ask questions and answer those questions. And so the idea is that we now have our genome assembly. What sort of questions can we answer using that genome assembly? And the most important thing that we can talk about, at least in evolutionary biology, is variation. What sort of variation leads to the varieties that we see out there in the world? And most of the time, there are lots of different types of variation, but most of the time we're going to be dealing with SNPs, or single nucleotide polymorphisms. These places in the genome where one base differs between me and you. Maybe I have a G at that position and you have a C at that position. There are other types of variation, but we're not going to worry about those right now. And so here's some examples of how you can do that. Basically, what you're going to do is you're going to take your organism and a bunch of organisms from that population or from that genus or from that group, and you're going to generate genome assemblies so that you can compare what sort of differences there are among those individuals. And you do that by doing what's called read mapping. You take your raw data and you map it back to your assembly. And by doing that, what you can do is you can identify things like heterozygotes. What you're seeing there is where this individual has A's at one position of the genome, but T's at the other position. This is where you would say, okay, from your father, you got a T at that position, but from your mother, you got an A at that position. And what you do is you would interpret this as saying, okay, well, about half those reads are A's and about half those reads are T's. That means that this individual is heterozygous. They have an A on one of their chromosomes, but a T on the other chromosome, and you would expect half of your reads to have an A and half of them to have a T. And so you can use that information. Whereas this individual down here at the bottom, it's all T's. So that means this individual is homozygous for that T at that position. It doesn't have an A at any place in its genome at that position. But you can also see where depth would be important, because how do you interpret this? There's only 4 reads at this position, and one of them says A, the rest of them say G. Statistically, that's really difficult to say whether that's a heterozygote or a homozygote, because we know there are errors, and that A could just be an error, or it could be a heterozygote. So what are some of the questions we can answer using genome assemblies and genomes in general? One of those things is to use something called GWAS, or genome-wide association studies, where what you can do is identify particular variants that are associated with particular traits, phenotypes, things that you can see. In this case, we're going to talk about disease. So here's the basic idea behind GWAS. You scan the entire genome, and you say, okay, we're going to focus in on this little thing right here, this little place right here, this one locus right here. In this case, that locus has a G at that position. And that individual is giving you the A-OK sign, I'm doing all right. And of course, he's going to pass that down to the next generation, and you're going to have a larger population. But at some point, maybe there's a mutation that occurs that changes that G to an A. That individual's still doing okay, but possibly in a couple of generations, you start seeing people get sick. And you say, okay, what's going on here? Is that A associated with the phenotype of getting sick? And it could be not. That's where statistics come in. And what we find is that this A allele is associated at 29% frequency with individuals exhibiting that sickness, getting sick with that disease. Is that significant or not? I don't know. Let's talk about an actual example. And this is going to be in humans because we don't have any, at least I couldn't find any examples in bats. Here's an example of something called age-related macular degeneration, in which you've got a whole bunch of people. You scan their genome using genome sequencing techniques. You compare those reads to the genome, and you say, okay, our null hypothesis is that alleles will be randomly distributed in the population, and there's going to be no statistical association with age-related macular degeneration. Of course, the alternative hypothesis is that there is some association. And we do that. What we find is that we can recover all the alleles from all these thousands or so individuals. And we find that, yes, there is an association of having a C at that position rather than a T, and it's statistically significant. There's a p-value of 1.2 times 10 to the negative 62 that, yeah, that C at that position in your genome seems to give you a higher risk of encountering age-related macular degeneration in your lifetime. And so this is something that we can do with genome assemblies and with raw sequencing data into investigating what phenotypes are associated with what genes or what alleles in the genome. Now, getting to something that we're all more interested in, let's talk about bats, because bats are cool. This is a paper that we published, I can't remember, a year or so ago, in which we were investigating the genus Myotis. If anybody knows anything about bat diversity, you know about Myotis. Myotis is crazy. Myotis is a very species-rich genus, and we don't know crap about it. They all look the same. They're all little brown bats. So my graduate student here, Jenny, wanted to look at Myotis and kind of get an idea of what sort of things were happening with regard to gene exchange in Myotis in the New World. And so she did this using genomic techniques. And her basis was this other paper that we put out several years ago, in which we generated a genome assembly or a phylogeny using mitochondrial DNA and using nuclear DNA. And we found that the two phylogenies don't match with one another. In particular, this species, Myotis branti, comes out at different places depending on what locus you use. And so she wanted to look at this. What's interesting about Myotis branti is it has an old world distribution. It's found in Asia and Europe. But yet, consistently, it is found amongst the New World species that we have here in North and South America consistently in both mitochondrial and nuclear phylogenies. And so we wanted to look at this and find out what happened. And using genomic data, we were able to say, okay, look, we got a really well-supported phylogenetic hypothesis that puts Brantai outside of the New World clade. It says, no, Brantai is an old world specimen, and in this phylogeny, it does come out as an old world specimen, and that's kind of what we expected. And this was using loci from all over the genome, unlike these other investigations here where you were using one locus or a multiple locus, but relatively few from around the genome. This was literally tens of thousands of loci located everywhere around the genome. So we expected to give us a more accurate picture of what we'd see as far as the phylogeny, a more accurate species phylogeny. We also identified introgression, which is the exchange of genes between species. In other words, hybridization events that you might find either ancient or more recent. And so we were able to investigate that. And then lastly, the last thing I want to talk about is this idea that we just, it should be accepted any day now. So I'm going to go ahead and talk about it. It was disentangling genetic diversity of Myotis septentrionalis in the face of white nose syndrome here in the United States. As you can see, we've got this species has a wide range, most of which has been affected by white nose syndrome. Here was our sampling regime across the United States. And what we found was that basically the entire species is one big genetic unit. It's called panmixia, where we have one set of, basically they're all interbreeding with one another as far as we can tell, with the possible exception of Louisiana, which seems to have its own set of alleles that other species don't have. And the idea here is now we know this genetic diversity within the species is high and pretty uniform across the entire continental United States. But more importantly, what this can serve as, in our opinion, is that it serves as a basis for future studies where we see what the impact of white nose syndrome is going to be as that population is devastated by that disease. So that's just 3 examples of things you can do. Here's a bunch of papers I just looked for yesterday on different things that could be done with genomics and have been done with genomics. Developmental questions, immunity questions, phylogenetic questions, population biology questions. These can all be addressed using genomic data. And so the take-home messages that I want to give you, because we got to wrap up, is that genomes are not the answer to every research question, but they can be really good tools. Genome assemblies are difficult to obtain. You saw that example there, but it's getting easier. And it's bioinformatically intensive. So to do bioinformatics, to do genomics, you have to have some bioinformatics skill, which is easy to attain these days. Most universities will give you that training. In order to get high quality DNA or genome assemblies, you have to have high quality DNA. So if you're working with Bat1K, we will help you to generate that high quality DNA so that we can help you with your project. And depending on what you want to do, there's probably a genomic answer or a genomic technique that you could apply to that. So I'm going to leave it there. This is the final picture of that puzzle that I was giving. And if you have any questions, I will be happy to do my best to answer them. I hope I didn't go over. Am I all right? Oh yeah, I can see my time over there. So does anybody have any questions? I see one in the chat. Brilliantly explained. Well, thank you, LR. So I don't hear anybody speaking. Is anybody left?
Oh yeah, we still have I think everyone except for one person had to go off to another seminar this morning, but there's still everyone here.
Okay. Well, anyway, does anybody have questions? Dana, as the host, you have to come up with a question.
Here we go. Here we go. Okay, so we have one. How long does the process typically take from DNA extraction to genome assembly?
It depends on the complexity of the genome. Because, for example, with something relatively simple like a bacterium, 20 minutes. We did a genome assembly on a Drosophila genome in my class last week. It took about an hour and a half. Once you get the sequencing data, generating the actual sequencing library takes a couple of days. Getting the data getting the DNA from the organism can take again a couple of days if you're doing a high quality extraction. So we're talking If you take the actual time associated with doing each step, it can take a week. This is very different from what it was many years ago when it would take potentially a year to do a whole genome assembly from DNA extraction all the way to assembly. One project I worked on for the Anolis genome, which is a little green lizard we have here in the States, it took years. But we did the Eptescus fuscus genome. We got the DNA, sent it off to Germany. They did the DNA extraction in a couple of days. The genome assembly took a week or so, and we're still working on the analysis. That's actually the hard part now. The hard part now is once you get the genome assembly, what, actually parsing out what it all means. So I hope that does that answer your question. I don't know if that was you, Dana, who asked that question or somebody else.
It was whoever LR is.
Okay. LR, how long? So, oh yes, that he said, he or she says yes, thanks. Any other questions out there?
All right. Well, I mean, that pretty much keeps us right on time with everything.
Good.
Oh, yeah, we just had another thank you. The presentation was excellent from Nathalie. So yeah, thank you so much, David, for being our presenter this month. And you really packed in so much information into that.
I didn't know what to expect. So
yeah, it was fantastic. Thank you so much.
I don't know if you want. I know this is going to be on YouTube. You're going to upload it there, but anybody is welcome to have these slides. They are.
Yep.
If you send me an email, I'm happy to share them with anybody.
We will make those available. We'll have the PowerPoint available, and we'll also be posting the slides as well. So
okay,
yeah. So if you want to come back to the information, feel free to do so. And again, just a couple more reminders for GBATnet in general is that we will be having our December webinar next month on the 6th, and we are solidifying our speaker for that over the next couple of weeks. And again, if you are a student or if you have students of your own, please consider sharing around the GBATnet student recruitment that is going to be opening up next week on the 15th. All right, if we don't have any more questions. Yeah, okay. Well, thank you so much, David.
All right, thank you, Dana. I appreciate it. And everybody else, thank you for attending.